Hi all, Dr. Clark here again. Today for Fish and Wildlife, we're going to talk about aging fish and wildlife. So we talked about capture, we talked about marking, and now we're going to talk about, well, what can you do with the organism in hand? Okay, and what some of the data is that you might want to collect and how that will allow you to make predictions about populations or, as a manager, better manage populations. And one way or one important piece of information is the age of an organism. Okay? Now, there are two types of organisms. There are organisms which we call indeterminate growers, and that means that they actually grow throughout their entire life. So they just keep growing. So things like fish, amphibians, reptiles, these things are indeterminate growers. They really never stop growing. Now, they definitely slow down. Okay? But their slow down period is typically because they shift resources from growth into reproduction. Okay? Now, those are indeterminate growers. And determinate growers are things like mammals and birds, where once they reach maturity or a little bit after maturity, the organism stops growing. It stops adding new length to the organism. It stops adding new bone material down, okay, and it concentrates on reproduction and maintenance. All right, so that being said, there are still ways at which you can age different organisms. It just happens to be that the way that you age an organism really depends on whether or not that organism grows throughout its entire life, or whether or not that organism stops growing at a certain point in time. Okay, so if we look at something like a fish or a fish scale, okay, so here you can see a fish scale. This is, what it looks like under a microscope after it's been pulled from the fish. Now, there are a couple things that I want you to note in this picture, okay, besides the things that are marked. Okay, you can see kind of a circle right here. This is called the focus, okay, and that means that's really the starting point of the scale. So when the scale started to grow, okay, it was, about that size, the center dot, okay? So that's the initial period, and then just like tree rings, material is being added to the outside of the scale or of the tree. Okay, you can see a different region here. You can see that there's kind of a line here, and this region here doesn't look nearly the same as this region. That's because this region here is the exposed region. Okay, now some scales, it depends on the fish. Some scales will be called ctenoid scales, okay, and they might have little teeny teeth or spines on them. There's scales like placoid scales. Okay, there's lots of different types of scales. But looking at a scale, you can tell where the scale has been exposed, and that's typically the side of the scale that looks very different, and you can't see annuli on them, or they're very faint annuli. Okay, so here's the focus. Okay? And like I said, fish are indeterminate growers. They grow throughout their entire life. Now, growth patterns really depend on food availability. Okay? And so, because most of the time indeterminate growers are ectotherms, their growth is directly related to the temperature of their environment. So in organisms that live in regions that have a summer and a cold winter, okay, then their summer growth is often much larger than their winter growth. because summer they're getting lots of food and they're applying that food into growth. Winter they're basically just living off of fat reserves and they have very little growth. It's just like an annuli in a tree. Okay? In the case of winter growth in a tree, the rings are very, very tight, and you barely see any growth. In the summer, spring, okay, you get large growth rings, a large amount of growth. Same thing in fish, okay, and we can see this in scales. Now, depending on the fish species, some species it's better to use scales. Other species it's very difficult to use scales, and it really just depends again on the fish species. But here you can see the focus, and then you can see annuli or growth rings that are coming away from the focus, and then you'll see a period, okay, where those growth rings stack up, okay, and they get really, really tight together, okay? And in actuality, you'll have some periods where the growth rings cross over, and we call these crossover marks. Those crossing over marks is going to be an indication that it's a winter or a slow period. Now, it doesn't have to be winter because you can do this in places that don't experience winter with the difference between breeding seasons and non-breeding seasons. So in a breeding season, so a breeding period, the fish will stop growing or have very, very little growth. And that's because they're applying all their energy into reproduction, just like kind of winter growth in certain, parts of the world. Okay? So it doesn't have to be summer, winter. It can be breeding periods, and it really is species specific. There are certain places in the tropics where fish might have 2 breeding seasons, so you might need to count 2 crossing over events for every year. Okay, but at any rate, most of the scale reading that we're going to talk about in this course is in temperate regions, in United States regions, away from the equator, where there is summer, winter periods, and you get these growth patterns. Okay, so again, this is summer growth, and then you have this band, this tight band. Okay, that's winter growth. Then you have summer growth. Then you have another tight band, winter growth. You have summer growth, another tight band, okay, winter growth, summer growth, another tight band, winter growth. Okay, so in that case, if we count those up, you have 1 summer winter, 2 summer winters, 3 summer winters, 4 summer winters. This organism is 4 years old. Now, the other nice thing about this, and I'll get to this in a little bit, is you can calculate. So clearly, when the scale was pulled, the organism was 4 years old. So you would have a size at 4 years old. But what if you wanted to know how big the organism was when they were 2 years old? Okay? We can do that. It's called back calculating or back calculating size. Okay? Because we know the relationship of the scale to the size of the organism, then we can back calculate and find out what the organism's size was, like when they were 2 years old or 3 years old, etc. Okay, and we'll come to that. We'll look at 2 different methods. One's called the Fraser-Lee method, and one's called the von Bertalanffy method. But we'll look at 2 methods to do that, to back calculate age. Okay, so let's look at some other things first, though. Why would you want age, or what does age tell you? Okay. Well, age can tell you quite a bit of information about your fish population. First of all, if we know that organisms, fish in this case, because I'm going to talk about fish, if we know that a certain fish species doesn't hit maturity or can't hit reproduction age until maybe they're 8, 9 years old, okay, then examining a age stock or egg structure stock will tell us, well, okay, so how much or what percent of the fish are in the bracket of mature and can reproduce, and what percent is in the bracket of immature and are not adding to the fish stock. Okay. Now, this might not just be fish. This might be the same thing if we're looking at birds, or if we're looking at mammals, if we're looking at amphibians, reptiles. Okay. The reason why I chose fish to discuss this graph is because, fish live for a long period of time. Okay. So 36 years or something like that. Okay. This is probably from, well, besides it says percent of fish, but this is from a fish group. Okay. If we were to look at something like, if we're to examine something like deer or elk or something like that, maybe their maturity mark is 2 or 3 years old, okay, depending on the species, okay, and they might only be living till they're about 10 years old or something like that. So the graph would be much smaller. Okay, but you can still get age, structure, and stock as long as you can age the organism. Okay? And that's where the different techniques of aging come into play. Is if we can age the organism, okay, and fish you can age per year. Okay? Other organisms like deer and elk and birds, you have to lump into groups of ages typically. Okay? But fish, you can age them out per year, and you can get a better kind of structure of their age. Okay, so here's another thing that you can examine. So maybe you're interested in figuring out, well, what fish are you capturing during different periods of time. Okay? So maybe you're interested in looking at, okay, well, what's being caught in the winter time? So what's the age group being caught in the wintertime? What's the age groups being caught in the summertime? And is there a difference? And by examining this, you can see most of the time what you're catching in the winter is 2-year-old and 3-year-old fish, with 4-year-old fish falling in. In the summertime, however, okay, it looks like you're catching younger fish. You're catching the young of the year, so the hatching fish, okay, that are not even a year, a bunch of 1-year-old, a bunch of 2-year-old fish, and then it starts falling off. So with very few 4-year-old fish caught, hardly any 5-year-old fish, not the case in winter. So this allows for researchers to figure out, okay, well, what's going on with the fishing season or with the harvesting season, okay? And at what point should we, calculate, well, what size bracket is 4-year-old fish, okay, and protect them in the winter fishery, because maybe this is a huge portion of your reproduction might be at this 4-year-old fish, okay? Or vice versa. Maybe there's too much take in summer of the younger fish, and there's not enough making it to the older sizes or older age and bigger sizes. So maybe we need to close down a lower bracket. Okay? So maybe a smaller size bracket is you have to throw them back, and the larger size bracket you can keep. Well, the only way you can know this definitely is by knowing the relationship between age and size. Okay? And you have to be able to age the organism to know that relationship. Once you have that relationship, then you really don't need to age the organism unless something drastically occurs in that population. You have drastic changes, introduce predators, you have climate change, a new competitor is introduced, poor habitat, something like that. That can shift age and growth around quite a bit. But as long as the system is fairly stable, you only have to do this really once. Then you have size, and you know what size range corresponds to a certain age, and then you can just examine the sizes. All right, so let's look at some terminology. So age groups is often, depending on what species we're talking about, what group of organisms we're talking about will use different terminologies for those different species. So 0 or first year of life is pretty common, okay, but you'll see when we talk about like birds and things like that, it's a little bit different terminology for those. The second year of life is often dictated by 1. That's a 1-year-old individual, and they're starting, their second year of life. Okay? Age classes, okay? It can be dictated by, okay, well, when were they born? Or when did they hatch? Okay, so what year? Okay, and so we can look here and we can examine something like sea bass, and we can see what their, what their populations have been doing. And so you can see this is a toe, so this is, like a net, probably a otter trawl or something like that, that's being pulled through the water and capturing, okay, sea bass during different periods of time. And you can start to see that you get, huge population spikes every once in a while. Okay? And you can note those surveys or note those organisms by either it's a spring hatch or a winter hatch or a spring collection or a winter collection. Depends on the species. Maybe these are examining fry in the water and you have a spring hatch and a winter hatch, so you have 2 breeding periods. All right, that will allow you to make predictions about populations. Right? We'll come back to a lot of this in a little bit. The other thing, the other thing that you can note is the cohort. So maybe you're sampling a population, managing a certain population, and you're interested in, well, how did that cohort do over the last 3-4 years? Maybe you made some alterations to the stream, maybe you made some alterations to their habitat by, planting trees or having tree rows or something like that. Or maybe the region's been clear cut recently or burnt recently. So you're interested in a cohort. Cohorts can be noted in a couple different ways. You can note them by the year. Okay? That's often what the cohort is. So you'll just say, okay, well, this cohort is the 2019 cohort. That means they were born, hatch, whatever, in 2019, and you're going to track them for a certain period of time. The other thing that you can do is maybe you're just interested in young of year, okay, cohorts across all years. So maybe you're doing a multi-year study, but you're only interested in the younger year each year. You don't really care what happens to them after that. So then you could just denote them as age 0, okay, or young of year. You could also note them as sub-yearlings, okay, so they're not quite a year old yet. Larval fish, there's lots of ways that you can note or distinguish the organism as an age 0 organism or that cohort. When we're talking about salmonids, okay, they're often called fry, and they're kind of has a size spectrum to it, 0 to 25 millimeters. Fingerlings are then from 25 to size at age 1. So with salmonids, the age 0 is broken up into 2 pieces. Okay, they're fry and their fingerlings. And the reason for this is because the organism has a drastic difference in life cycle or lifestyle, okay, between being a fry and a fingerling. As a fry, okay, at least for a good portion of the fry's life, they're feeding off of a yolk sac. Okay? As a fingerling, there's no more yolk sac, and they're feeding on, typically aquatic invertebrates in that environment, okay? And, they might be migrating also away from their hatch grounds into a different region. The other thing that I can say is some larval fish, and it's not just salmonids that get the term fry and fingerling, okay? So there are other larval fish that get the term fry and fingerling. It was first denoted probably for salmonids or the salmon, trout, those groups. But now it's used often by managers to denote, this size fry. That's, basically the organism is just hatched, and then fingerling is after hatch and after the yolk sac's gone and they can feed on their own. That's typically noting the difference between the two organisms. So again, back to salmonids. Hey, you can see salmonids have a distinct feature that differs between different stages. So you can have in salmonids, you can also at that fingerling stage, okay, you can also call them par. Okay, and par denotes these little dark markings, okay, that run the length of the lateral line, so that you can see. So you can ignore the red spots, okay, that's species specific, but you're going to have this lateral line, this line that runs down the fish, and you're going to have par marks, which are these black spots that run along that lateral line. Okay, majority of every salmonid has par marks, okay, when they're in that fingerling stage, okay, or in the par stage. Okay. Those par marks will eventually vanish as the organism, as the fish, goes through smoltification. Okay? So smoltification is going to be adapting to a different environment. So moving from a freshwater system into a marine system, a saltwater system, the organism adapts physiologically. Okay, their kidneys change the way they react to salts. So in the case of freshwater fish, because the system itself that the organism is living in is freshwater and has less dissolved solutes, dissolved salts in it, the fish itself will need to drink water because, or will need to urinate often, okay? Basically constantly, because if the outside system, and this goes back to general biology, which majority of you will remember, okay, if the outside system is freshwater and the inside organism is more salty, has solutes, then water will want to run into the organism. It will want to be absorbed into the organism, into its cells. In order to rid itself of that massive amount of water that's constantly coming in, freshwater fish have to urinate or, dispose of that excess water constantly. Okay. Well, when the organism then switches to a marine environment, now the organism's body has less salt than the outside system. So any water that's in the organism is going to be rushing out of the organism, out of its cells into the environment. So in a marine environment, okay, the fish has to constantly drink water, constantly take in fluid, excrete salts as much as they can, okay, or hold salts in the kidney to keep their salt level up so they don't rid themselves of water as quickly. Okay? And that's part of that smoltification process. Well, during the smoltification process, those par marks go away. That organism gets this kind of shiny new scaly or scale system on it, and its kidneys and internal body kind of switches over to adapt to marine life or the marine environment. We all know, well, most of us know, what then occurs As those organisms move from the marine environment back to a freshwater environment, okay, majority of species do not have the ability to switch their system again. So as they enter a freshwater environment, water is rushing into their cells, okay, which causes their cells to rupture. The organism starts to develop sores, and the flesh really starts to just basically fall off the organism the more time it spends in fresh water because the adaptations that it no longer has for that freshwater environment. Okay? The other piece of that movement into a freshwater system is majority of salmonids are kind of one and done spawners, one and done reproductive events. So they sink all their energy into reproduction for one event, okay? Which means that they don't have the capability to maintain cells and tissues and things like that. So the organism starts to break down. Okay, other denotions of different cohorts could be immature, juvenile, subadult, adult. This is very common when we talk about birds. We might also call the organism pre-breeding, okay, post-breeding, just ways to note different periods of time. Okay? And this is very true for organisms that it's hard to distinguish their actual age. If you can't actually age the organism, or the organism has to be dead and you have to, examine their bones to age the organism, then often we'll note them as post-breeding, pre-breeding, immature, juvenile, subadult, adult, that kind of thing. The other notion can be different stages within the nest. Maybe they still have their natal down, okay, and which is kind of the fluffy feathers, the warm feathers that a lot of organisms are going to be born with to keep them warm during different periods of time. Okay, so here you can see that kind of fluffy down here on this bird. All right, the other notion that is important is the difference between, especially in the case of birds, is the difference between life cycles in birds. Just like we have differences in life cycles in fish, okay, whether you're inamorous and can move, or you're moving from marine environments to freshwater environments, whether you stay in freshwater, stay in marine environments, okay, is important to know. Same thing with birds is whether you're a precocial bird, okay, so a bird like a goose or a duck, okay, where you hatch, you have this natal down, but the organism at hatch has the capability of feeding on its own, has the capability of basically living on its own right at hatch. Or are you a bird that's altricial, okay, where you're born nearly naked? You will have some feathers on the organism to keep them a little bit warm, but the warmth really comes from the nesting material and the parents to these offspring. Okay, depending on the species, it could just be the female. In other species, it's both male and female that will, keep warmth on the birds as they're developing. Okay. Also, species specific, whether or not the bird is completely blind at hatch. Okay. It really depends on, how advanced the stages are. It really depends on the bird itself. Okay. But again, don't have the capability of feeding on their own. Okay. They're going to call to a parent, and that parent will bring them food. That's altricial. Okay. Precocial birds, again, they might follow the parent around for a long period of time for protection, okay, but they feed on their own. The warmth comes from the organism itself, and they don't really require extra energy from the parents. Okay. Now there are some major distinctions between precocial birds and altricial birds that can come into effect with this. Precocial birds often are a much larger egg, or hatch from a much larger egg, have a lot more energy in that egg, and often will have a longer incubation time. Okay? Altricial birds typically come from a smaller egg, less energy, hatch quicker, okay, but then spend a longer period of time in the nest, begging for food, that kind of thing. Okay, and it really just depends on the species of bird, which way the organism is going to go. Okay? So there are other things that need to be noted from a bird perspective. Okay, when we talk about plumage, we can talk about juvenile plumage. So you can see here a couple ways to note that this is a juvenile. Is you can see the natal down, lots of it, but you can also see that none of the primaries on this bird are completely unsheathed. So you can see that blue kind of or bluish white kind of material there. That's the sheath that goes over the feather. Okay? And that organism has not developed its primaries. Its primaries have not been unsheathed. So that would indicate that this is a juvenile and the bird can't actually fly. Or if it can fly, it doesn't fly very well. Hatch year is a way, and we talked about this in the banding video. Hatch year is a way to denote, okay, that organism hatched this year. It's not mature enough, and maybe it's already molted. Okay, maybe it's moved into its adult feathers. Okay, but it hasn't spent an entire year. So this is still noting that this organism's not one. So it would be an age 0 organism. And you would indicate that by saying it's hatching year or hatch year. Okay. The other ways that you can examine the organism, you can examine the organism's tail or feathers, okay, to distinguish whether it's hatch year or non-hatch year. Okay. A lot of juveniles that have not molted their feathers yet will show the inside track might be thinner, smaller. Also, their feathers typically come to a point versus being kind of squared off. And this can be true for tail feathers and primary wing feathers. Okay, adults will have kind of a squared off or a sharper edge to their feathers, and I'll show you this hopefully on a live bird in some of the misnetting videos, but you can see here that this comes to a point, okay, or kind of is V-notched, okay, in that this is a juvenile's feather, okay, tail feather. Adults will be more round and then have a sharp edge coming off, and you won't have that feather won't come to a point. Okay, and so to distinguish that it's not a juvenile, so it's not in juvenile plumage and it's not in the hatching year, okay, we would note either via feathers or other ways like it's sitting on eggs or something like that, that this is after hatch year or AHY. Okay, now you can do this by examining, and it really depends on the species, okay, what you're going to be examining. Okay, majority of species you can examine the primary feathers to figure out whether it's after hatch year. So here you can see this feather is rounded and then it comes to a sharp kind of distinguished ridge, okay, or straight line. Okay, that indicates that this organism is after hatch year. It's already molted its feathers once, okay, and it's in its adult primaries. Okay. There are other ways that you can do this. You can examine secondaries. Sometimes the bird will have different colored secondaries, okay, or different colored plumage. Okay, in the case of maybe the natal down is different colored, the tail feathers are different colored as a juvenile, the coverts or the wing covers or the primary covers are different colored. There are lots of different ways, very species specific ways to determine whether it's hatch year or hatching year or after hatch year. One key way, though, is that sharp ridge or that straight line that comes off of a rounded tip on the primary feathers. All right, so let's look at some other methods outside of birds. Let's look at actually aging organisms. So again, birds are not indeterminate growers, they're determinate growers, so they stop growing basically at maturity, okay? And the organism's about the same size. Now, if you spend enough time with birds and studying a certain species of birds, you might have a size breakdown of, okay, that's a 2-year-old bird, or another size breakdown, that's a 3-year-old bird. Okay? But it's still difficult to age the organism. So that's why I said when it comes to birds, it's either hatch year, they're either a juvenile or they're an adult. Okay? When it comes to aging other organisms, you can age them using hard parts. So you can age fish using scales. Again, depends on the fish species. Some species are better than others. You can use fish by using otoliths. Otoliths are ear bones. Okay, so inside the fish's head, they have a labyrinth. Okay, and they have 3 otolith bones. Okay, the sagittal, the asterix, and the lagital. Okay, so they have 3 ear bones. Most species, you're examining. If you're examining otolith, you're either examining the sagittal or asterix ear bone. Okay, otolith. Well, anyways, those bones sit in a fluid-filled sac. Okay, and it's like a gyroscope. So that fluid-filled sac. Anytime that bone moves inside that fluid, the fish knows that it's moving, the direction is moving, okay? And so it knows which way's up, it knows which way's left, right, and it's just a way for the organism to right itself. Okay? But again, these are hard parts. The nice thing about using otoliths versus fish scales can be damaged by the environment. Okay? So, a fish is swimming through an environment, it can get, scuffed up. Fish regrow their scales, so if you remove the scales off the fish, the fish will regrow those scales, and regrown scales have massive fast growth, so you won't be able to age the organism using a regrowth scale because that scale is playing catch up. So instead of having, a steady growth rate over years and years and years, uniform growth, if the scale is regrown, that scale, a lot of energy goes into it to play catch up, so the scale covers the fish in that region, and you can't distinguish whether it's a regrown scale or not. And there's equal protection from that regrown scale and a non-regrown scale. Otoliths, on the other hand, are inside the fish's head, and they don't suffer kind of damage from that point of view from the environment. Okay, so here you can see different otoliths. Okay, you have the sagittae, okay, the asterix, the lapilli. Okay, like I said before, most fish you're using the sagittae or sagittai, okay, and the asterix or the asterix. Okay, very few fish are using the lapilli. Okay, they have 3 on each side, so that's why you see 6 otoliths here. Okay, and just like the growth rings on the scales, same kind of deal. In the fish, you have in the otolith, you have a focus, you have a center point. Okay, and then the growth rings or the bone material is being added onto the outside of the material, outside of the otolith, and you have a slower growth period, okay, and a faster growth period, okay, just like you have summer, winter. Okay, and like I said, it really depends on the fish species whether this is summer growth, winter growth, or whether this is reproductive period, non-reproductive period. Okay? The other thing that you can use, and often is used in things like sharks, is a centrium. Okay, so another nice uh, bone to use. But again, just like the otolith, the centrium, these organisms, if you're going to use otolith and centrium, the organism has to be dead. You have to sacrifice the organism in order to age them using those material. A scale can just be pulled out. Okay? The fish will regrow that scale. The fish doesn't have to die. It's a really simple method. Okay? Centrium, you can see here, centrium is part of the vertebrae, that circular part. And you can see you have a focus, and then you can see you have rings that are running along that or out of that centrium. Okay? This is often used in things like sharks and skates and other chondrichthyes or cartilaginous fishes is often used this because their scales are placoid scales or teeth scales, and they don't show those growth marks nearly as well as, say, a centrium. Okay. Now, some of you are probably fairly familiar with aging deer, elk, ungulates, this kind of thing, by using their teeth. Now, this is a good technique as long as you know something about that organism and that organism's diet and where that organism is spending the majority of its time. Okay. Now, early on, it's really diet and everything like that don't play a huge part. Okay, because it's just like other mammals. You have kind of baby teeth and non-baby teeth. So you have premolars and then molars. Okay, in a fawn, you will only have one molar. Okay, the other ones will have not come up yet. Okay, even in a year and a half old deer, you might see all the molars, but they're not completely cusped. So the third cusp has not came out yet. Okay. After that year and a half, moving into the second year, then you'll see that third cusp. Okay, all the molars will be present and the premolars will be present. After that point, so after 2 years, then it really gets into how much wear and tear has been occurring on that organism's teeth. Okay, so now you're looking at how much enamel is present, how much dentine is present. Okay? And you're looking at kind of the ratio or the amount of cuspness or how sharp those points are in the organism's mouth. Okay? Now, like I said, this really depends on what the organism's diet is. If the organism's in an environment where it's eating a lot of very hard wood, okay, so it's browsing a lot, then that dentine and enamel can be worn off much quicker. Or maybe it's in a sandy environment where it gets a lot of sand and things like that that can grind the teeth down in that organism's mouth. Okay, versus maybe an organism that's feeding on alfalfa or grasses, and that's all it's eating. Maybe their dentine and enamel last much longer. Okay, and you don't get that wear pattern. So again, like I said, up to about 2 years, this is universal. After that point, you really need to know what environment this organism is coming from. And that will kind of dictate, all right, well, how much wear and tear can you expect on the teeth? Okay. And so, but you have the capability of, again, analyzing these organisms via their teeth. Okay. Not all the time, but most of the time, this has to come with the organism being deceased or dead in order for you to get a good analysis of whether it's a 2-year-old, 3-year-old, 4-year-old, 5-year-old organism. Okay. There are other ways that you can do this. Okay. In other mammals, you can take a tooth and you can section the tooth. You can actually cut right through the material. You can do a cross section of the tooth, okay? And you can count rings, okay? Because these organisms are not losing their teeth, and they continuously grow their teeth for a long period of time, okay? So predators are often, teeth are often used, okay? In things like amphibians, bones are used, normally digits, fingers, okay? And so there's a common marking technique that we talked about before. Amphibians and reptiles, this is a case, where you clip toes in order to mark individual organisms. Well, those toes, those phalanges, can be utilized then by doing a cross section and reading them just like a growth ring. Okay, most of the time they have, kind of marrow or some kind of blood that's in the inner region. Okay, but you can read it moving out. In some other phalanges, you have to do a cross section and a transverse plane, and then you read it, okay, just like you would here. But again, you're reading growth patterns, you're reading new material being added on, and then a period where that material kind of stops being added or it slows being added, and then a speed up period. There are other ways, okay, in things like organisms that don't lose their horns, okay, so bighorn sheep, okay, these organisms, you can read the rings, okay, because they'll have fast periods and slower periods, and you can get a good basis to how old that organism is. It might not be perfect, okay, but you can tell definitely whether it's a 1 or 2 year old organism versus a 5 or 6 year old organism. Okay. Other things, you can look at length of digits sometimes, or you can look at whether or not the organism has reproduced, and if you know at the age the organism reproduces, okay, then you can get an idea of, okay, this organism has already reproduced, so we know that it's older than 2 years old, okay, and then based on digit length or something like that, maybe you can work it out to 4 or 5 years old, 6 years old, whatever it might be. Okay, so now let's look at back calculating size at age. Now, the reason why I spend most of my time, or I'm going to spend most of our time talking about fish, is because fish are the organisms that are most likely going to be aged to a specific year. And in fact, in some fish species, not only can you age them to a year, you can basically age them to how many months old are they, and in some, even maybe how many weeks old they are. Okay? So fish really, their hard parts really give us this unique opportunity to kind of age these guys out. Okay? But let's say we want more than that. We don't want to just know how old they are. We want to know what was their size at a given age. Okay, so let's work through one of these using the Fraser-Lee method. Okay, so this is a scale. This is a tenoid scale. You can see the teeth on it here. Here's the focus. So at 0 years of age, we're saying that it was 0 millimeters. We'll come back and you'll see this is what C is. And we'll see that it's not actually 0 millimeters at 0 years of age, because that would say that, when the organism hatches, that there's no size to it. Well, we know that there's actually size to it, okay? But right now, I'm just putting 0 millimeters because I don't know what the size of that organism was at 0 years of age. We can calculate that, and I'll show you how in just a second. Okay, at 1 year, or at the annuli of 1 year, the distance from the focus to that 1 year mark is 2.3 millimeters. Okay, I measured to 2 years is 4.6, 3, 5.4, 6.1, 6.6. Okay, so now I want to know what the size of the organism was at 2 years of age. Okay? I know how, how big the scale was. It was 4.6 millimeters from the focus. Okay? Knowing that, I can use the Fraser-Lee method to back calculate the size of the organism at 2 years of age. Okay, so what is the Fraser-Lee method? Okay, so we have to kind of use this equation, and this is what we're after, Li, or the length at a certain age I. Okay, in this case, this would be L2. We're interested in the age at 2 years of age, or the length at 2 years of age. Okay, LC is the length of the organism at capture. Okay, we already knew that. Whoops. I said it was 690 millimeters. Okay. SI is a scale length at the annulus eye. Okay. So how big from the focus to the point on the scale of 2 years old, how big was that? Or what's the size of that? In this case, it's 4.6 millimeters. Okay. SC is the total scale length from the focus all the way to the edge. And in this case, it's 6.6 millimeters because 5 years is right at the edge. Okay. C is the intercept from a regression of scale length versus body length. Okay. The only way you will know this is if you've caught enough of these organisms, enough of this species of fish, and pulled enough scales off to measure the scale and the fish. Okay. Well, I've done that. Okay. And here you can see this is the scale length on the x-axis. On the y-axis is the fish's length. And I fit a regression line between those points. Okay. So I just plotted these on a scatter plot, fit a trend line, okay, a straight line through those points, okay, and I have an equation. Okay. Right here is where that line actually crosses the y-axis. Okay, that's my intercept. And it says it's 24.824. That's my C. Okay, 24.824 is C, or the intercept. Okay, so at age 0, the fish is hatched already having a size of 24.8. Okay, so I can utilize that, and I'm going to. Okay, so 24.8 millimeters. Okay, so now plug it into the equation. 24.8 millimeters, C. 690 millimeters is my length at capture. 4.6 is the distance to the second annuli. Okay, and 6.6 is the distance from the focus to the edge of the scale. By knowing that, I can back calculate the age of this fish. At 2 years old, this fish was 470 millimeters, give or take some. Okay, but that's really neat, and that's a really great way to find out, well, what happened in this environment with a new introduced predator, or a new prey species, or a dam was put in, or the water was, converted and moved, okay? Or we had a huge drought during that period of time, or we had a fire that ran through. Well, let's say a fire ran through, you caught this organism 3 years prior to that, a fire ran through. Now you know, okay, well, this organism was 2 years old. It had great growth first year, second year, and now the growth is stunted, okay, in the last 3 years. Is that due to the natural growth of the organism, or is that due to this organism having some kind of environmental effect? And that's the nice part about using methods to back calculate things. Okay? Fraser-Lee is a really simple method. Okay, there are lots of problems with it, but I want you guys to know how to use the Fraser-Lee method. There's another method which is called the von Bertalanffy growth equation. Okay, it's same kind of premise, except for it gives you a more natural curvature to the growth of an organism. we know that even though these organisms, because you're applying this mostly to fish, that these organisms are indeterminate growers. We know that. But that doesn't mean that they're not going to kind of putter out or kind of hit a certain size, maximum size, because they're going to, like I said before, convert a lot of that growth energy into reproduction. So you use von Bertalanffy growth equations really to get at, okay, well, what does this organism look like, okay, over its lifetime? Okay? What are these patterns? Okay? And then if you know these and you've created a really good von Bertalanffy growth equation, you really don't have to age the fish anymore. You can just say, okay, I'm going to look at the total length. Here's the total length in centimeters. Okay, let's say it's 40 centimeters. I come over, there's 40 centimeters, but we want it where it intersects with the graph, right about there. Come down. This is about a 7-year-old fish. Okay, and by creating these you have to create them in basically each waterway. Okay, but once you have it and there's not huge alterations to the environment, and like I said before, no more aging fish. You don't have to crack otoliths and process otoliths or read scales or anything like that. You just get the length of the fish, fit it to your von Bertalanffy, find out what the age of the organism is, and go on your way. Okay? So again, that's one of the reasons, and a couple of the reasons why you would want to know the exact age of an organism. Okay? There's lots of other reasons, whether it be reproductive output. So again, we talked about this before with that brook trout example, with 1-year-old, 2-year-old fish reproducing most of the offspring, 4, 5, 6-year-old fish, okay, producing very little. Well, you need to know that. Well, you have to be able to analyze the age of the fish, okay? And it might not just be directly related to size in some fish. Okay, this curve might curve a lot more where they hit their maximum size a lot quicker, okay, than this group of fish that's shown here. So the age is really meaningless, except for it's telling you how many reproductive bouts it's had, not really its size. Okay? Well, with that, we'll come back and we'll talk about reproduction, and we'll talk about sexing organisms, because not only do you want to know what the age of the organism is, it's also important to know what's the sex of the organism, because that will tell you what's your sex ratio. That might tell you what your growth rate is, because it might be determinant whether it's a male or a female, and that can tell you a lot more about your population. Okay, so next time we're going to talk about sex of the organism, or how to sex organisms.